This is a little video about ribosomes, and ribosomes look amazingly complicated when you see them in diagrams. You just need to go on Google a few images, and you can see that they come in a sort of huge variety of what looks like different shapes. Effectively, if you sort of blew it up big enough and looked at the sort of how the protein and RNA are wound around each other, it would look very complicated like this. However, you don't need to know that. You need to be able to recognise them. But what you're effectively looking for is no matter how many fingers they've got, so some of them are sort of drawn sort of almost like that with another unit across them, we can simplify it right down. They are a big subunit called the large subunit and a small subunit. So they look a bit like a snowman upside down. And sometimes you'll see them with a little molecule of messenger RNA lying across them like the snowman's scarf. You'll see why later on in the course. So messenger RNA, you remember, leaves through the nuclear pore. And you'll remember from the video on the nucleus that ribosomes are made by the nucleolus. So they've got a large subunit and a small subunit. Now, the other thing that we know about ribosomes is that they come in two forms. We've got 70S ribosomes and 80S ribosomes. 80S, these are larger, and again you can find out if you're interested on Wikipedia or anywhere, and these are associated with eukaryotes. And the 70S ones are smaller, and they're associated with prokaryotes. The S stands for Svenborg unit, apparently. I always thought it was sedimentation, but perhaps not. So smaller, larger for eukaryotes and smaller for prokaryotes, but remember also these lovely little small ones providing evidence for endosymbiosis in mitochondria and chloroplasts. So that's one of the sort of similarities, if you like, between the mitochondria, chloroplasts and prokaryotes. So large subunit, small subunit, sometimes you'll see them drawn as a sort of a big triangle and a little oblong. That's one of my favourite ways to draw them. But just remember, large, small subunit, look a bit like a snowman. So where do we find them in the cells? Well, in eukaryotic cells, where we've got membrane-bound organelles, we will find them bound up onto the membrane of the rough endoplasmic reticulum. So this is rough endoplasmic reticulum. We've got those membrane cisternae with our little ribosomes sitting like a row of snowmen on top. We also find them free in the cytoplasm, so these are between the cisternae of the endoplasmic reticulum, and these little dots are the free ribosomes. So ribosomes are teeny tiny. They're the smallest thing that you're going to see, I think, on an electron micrograph, and they just look like little dots. You can't really distinguish between the large and small subunits. So we need to deal with their function. They are really concerned with protein synthesis. So we haven't really done messenger RNA. We'll do it much later in the course, towards Christmas. The messenger RNA actually carries that DNA code out of the nucleus through the nuclear pore. It attaches to the large, in between the large and small subunits, remembering that these two things are themselves made of RNA, ribosomal RNA, rRNA, and protein, and they form a place where a different sort of RNA can bring down the amino acids into the right order, and that is called transfer RNA, and we'll be doing that in detail. So main job in everything is to manufacture protein. So we can see now that that links to the rough endoplasmic reticulum manufacturing and transporting protein. Where is it being transported to? Well, to the Golgi body. What's it going to happen there? It's going to be modified and budded off into vesicles for lysosomes, secretory vesicles, or extramembrane.